Okay, so hello everyone and welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Swathi Kiran. Dr. Kiran is a James and Cecilia Ying Professor of Neurorehabilitation in the Department of Speech and Hearing Sciences and the Director for Strategic Implementation Planning at Boston University. Dr. Kiran examines language treatment for individuals with aphasia, bilingual aphasia, and neuroimaging of brain plasticity. She has over 120 publications in high-impact journals of cognitive neuroscience, neuroimaging, rehabilitation, speech-language pathology, and bilingualism. She is a co-founder and scientific advisor for Constant Therapy, a software platform for rehabilitation tools after brain injury. I would just like to remind the audience to please address any question you have using the Q&A box or just raise your virtual hand. Dr. Kiran, thank you very much for coming here today. The virtual stage is all yours.
Thank you, Or. It's good to be back at this group again. I gave a talk several years ago now when it was pre-pandemic and we were still at the Martinos Center, but it's nice to be back. So let me get started. Are we ready?
Yes.
Okay. So my first slide, I'd just like to get out some disclosures out of the way. As Or mentioned, I do have a significant financial conflict of interest with Constant Therapy as I'm an advisor and I own stock. I've also been fortunate to receive grant funding for all the work I'm going to report today from NIH, as well as the Coulter Foundation and Asher Foundation. Okay, so just getting started here, let me introduce you to the topic today. It's acquired brain injury. And if you didn't know this, acquired brain injury is a broad term that includes either a stroke or a traumatic brain injury and hampers people from communicating effectively and successfully and participating in their lives. There are many factors that determine the degree to which someone with an acquired brain injury will recover their function. Among these are biological factors such as the degree to which neuroplasticity, such as the changes in dendritic spines and connections occur. Also, individual-specific factors such as the degree to which initial severity, age, the lesion size, the location of the lesion, all that influences the recovery as well. Finally, the kind of behavioral interventions or the treatments that adults with ABI can receive, including the intensity of those treatments, the salience, the kind of specificity of the treatment also determines the degree of language recovery. So these are all things we're going to talk about today. Here's sort of the roadmap for our talk, for my talk today. The work at Boston University so far has focused on teasing apart these factors of that influence the recovery. I've sort of lumped it into three buckets that I'll be focusing on today. The first one, the first one is looking at brain structure and function, so neural markers of recovery. The second bucket is looking at language and cognitive markers of recovery. We've spent several years examining the nature of language and cognitive impairments that influence the inability to function successfully in the environment. I will not talk much about this at all because it's a whole different talk and I'll just sort of give you the high level overview. Finally, we've spent many years also developing effective therapies, rehabilitation for specific language and cognitive function deficits. Again, I won't have time to talk through all of them, but I'll give you enough context for where we're going today. But more recently, we've been trying to integrate all these pieces of information, these multidimensional factors, to develop machine learning and computational algorithms to predict which of these factors are important in predicting recovery and how these pieces of information can help us better predict treatment outcomes for our patients and give them a prognosis that's individualized and that's personalized. So this is sort of at a high level, the focus of my talk today. Okay, so a lot of today's talk will really focus on neural markers because that's what this audience is tuning in for. And let me start with a very high level sort of update. If you haven't been keeping up with this field, what's happened in this field so far? This is a schema that essentially combines a bunch of studies. On the x-axis is the time of recovery after a stroke, and on the y-axis is the amount of brain activation that's been put together from different studies. Um, the There's a summary by Hillis and Heiler who proposed a three-stage model of spontaneous recovery, where in the acute phase, which is in that blue box on the left-hand side, which is really in the hours and days after a stroke, what's really important is the reinstatement of the function of the ischemic penumbra, so the tissue around the stroke. And so reperfusion of the ischemic penumbra is key. In the subacute stage, which is weeks and months after the stroke, which is in that gray box, reorganization of brain structure function relationships are really important. So to what extent the brain can reorganize and reconnect. And then the chronic phase, which is months and in years post stroke, establishing new pathways and developing compensatory mechanisms, those are really important. Now, in this, overlaid on this is a key study by Dorothy Sauer that will be relevant, where they looked at the degree of activation over the course of 1 year of spontaneous recovery. So the red line here is what healthy people show in terms of brain activation. The dotted line shows you the left hemisphere activation, which is reduced because of the stroke. But over the course of the year of recovery, this activation returns back to normal. What's very interesting is that this other dotted line that shows the right hemisphere activation actually shows an upregulation during, as part of the course of recovery in the subacute phase, but it's then followed by a normalization or a consolidation of the language system in the chronic phase. So the point here is that it's fairly dynamic in this early stage of recovery. For today's talk, I'm really going to focus on this large, big blue rectangle here, which is the chronic phase, which spans months to years after the stroke. Although physiological changes occurring during the repair phase are generally subsided and the brain is sort of in a stable state, the mechanisms facilitate plasticity remain at play and are adaptable to the kind of treatment and environmental experience. So throughout the lifespan, the brain is changing. And in the case of stroke survivors or acquired brain injury survivors, treatment can result in such changes. So that's sort of what we're going to focus on. And I'm just going to give you the story up front, and then I'm going to try to make the case for the story along the way through my talk. What I'm going to try to convince you, or at least show you evidence for, is this high-level framework that we're collecting data in our lab for. And this relates to this model of behavioral recovery in post-stroke aphasia. This is, we're looking at left hemisphere stroke patients in this particular model. And over the next set of slides, what I hope to show you is that recovery from aphasia is very individualistic. No one size fits all, especially in the chronic phase. Recovery to normal function can really depend on the degree to which individual brains have reorganized networks and network connectivity. So really the hypothesis here is that patients with efficient, better network typology will show greater improvements than people with inefficient typology. So I'll come back and explain this slide later on once I've shown all the data. And also, just as an overview, I will be sharing data from a range of methodology that we've conducted in our lab that includes traditional fMRI and DTI work, but it also includes more recent work with FNIRS. So it'll be a range of methodology here. Okay, so let's get started. Back in 2016, in our first study that really tried to look at this question, we had 14 patients with aphasia, and here's their lesion overlap. And we were trying to understand what brain regions were active when patients were doing semantic processing tasks. So we identified specific regions in the left hemisphere, right hemisphere. And in the left hemisphere, we were looking for the degree of damage to the degree of correlation with other regions. And the first finding was that the more the damage to the left hemisphere IFG and middle temporal gyrus, angular gyrus, supramarginal gyrus, the higher the percent signal change during a bold fMRI task of semantic processing. But the higher this change in bilateral superior frontal gyrus, middle frontal gyrus, and anterior cingulate. So the first conclusion we drew from that paper was that when you have damage to these left hemisphere frontal regions and temporal regions, there's actually an assistive network in the bilateral frontal regions that help in. And then we looked at signal to signal correlations. This was before we started doing any network analysis. And what we found, as we would expect to see, is that when we looked at percent signal change correlations between regions, controls showed a left hemisphere lateralized network. Mostly, these were all positive correlations. But the stroke patients actually were much more bilateral and to a larger extent frontal right hemisphere as well. So that's what you are seeing on this side. Let me see if I can use my pointer here. That's what you see on this side of the brain. And there were very few negative correlations, but there were some between regions as well. So this was the first study where we thought, we sort of realized that brain activation in stroke patients is not just left or right. It was sort of a combination of both hemispheres, and it was much more of a dynamic relationship than we had thought it out to be. So from that point, we started looking at what other aspects of the brain structure and function might influence the recovery. So again, this is another study. We looked at 30 patients who had a stroke. They all underwent T1 and DTI scans. They also did fMRI. We'll come to that in a second. But they also, they received a treatment that I'll be talking about much later on. But in this study, we tried to look at these lesion information more specifically in terms of gray matter metrics that included the amount of spare tissue within 5 cortical regions. And you can sort of see that in this picture here. We also calculated white matter integrity, which was calculated by the degree of spare tissue and fractional anisotropic FA from 4 homologous left and right association tracks. And then we asked the question, is gray matter information enough, or do you need a combination of gray matter and white matter information, the integrity of this information to predict to what extent aphasic patients will recover language and recover from treatment. What we found was that for overall aphasia severity, so how severe their language skills are, we found a significant correlation between aphasia quotient, which is a measure of aphasia severity, and all left hemisphere gray and white matter metrics. But this was when we did not control for lesion volume. And this figure actually, without getting into the details, is trying to do a dimensional analysis between lesion size and location. But when we did control for the overall lesion volume, everything disappeared. So overall lesion volume was really explaining all of the aphasia severity in our patients. But then we also looked at, so that's what I'm not showing you the data for, but we also looked at naming scores, and we found that there were significant relationships between the white matter tracts integrity as between language function and so these connections. We also looked at something called proportional maximal gain. This is a treatment outcome measure that I'll talk about later on, and this is how much you can improve from treatment. Then again, there are significant relationships between specific tracts like the left IFOF and left ILF with the PMG. But the high level story here was that preserved left hemisphere regions predicted language severity and treatment outcomes, but only after, to a lesser extent after we controlled for lesion volume. But both gray matter only and gray matter white matter clustering predicted baseline aphasia severity and naming skills. That's the Boston naming test. But only gray matter plus white matter clustering predicted treatment outcomes. So something about the base integrity of the brain regions and their connectivity predicts the extent to which patients will improve from treatment. But I'll come back to this story again and again. And so from that point, so that was sort of looking at structural integrity. We also wanted to see to what extent brain regions communicated with each other when they were performing in these individuals with a stroke. So in the next set of studies, I'll talk about fMRI data. Patients here, this is a small initial study that we did back in 2016. There were only 13 patients. We're having them do a picture naming task inside the scanner. So they either say the word belt or they say the word skip. And we're looking for connectivity. This is looking at dynamic causal modeling just between 3 regions. We picked the middle temporal gyrus, inferior frontal gyrus, and middle frontal gyrus as our regions of interest in this connectivity modeling. And then we designed this dynamic causal modeling space where we build all plausible connections between these regions and how they might be functioning in these stroke patients. And long story short, what we found was that the models with input to the left middle frontal gyrus best fit with the PWA, the patient's data, which is a bit surprising. because we don't expect middle frontal gyrus to be traditionally involved in language. But we also found positive correlations between spared tissue and behavior and task effects to regions, and negative correlations and between the coupling of regions that I'm not showing you right now. But the bottom line here was that even though we traditionally think of left inferior frontal gyrus and MTG as critical brain regions involved in something like picture naming, the left MFG was actually critical in the recovery of the language network in our stroke patients, as you can see from this graph right here. So this was looking at picture naming. We did another study, which was essentially to replicate and follow up with a different task. The patients here are doing a semantic feature naming task, semantic feature verification task. We built exactly the same model, the same dynamic causal modeling. The task is different, and we ran it with patients and controls. And again, similar to picture naming, models with driving input to the LIFG best fit the control data as it was in the previous slide. But for patients, the best fit model actually was to the MTG. Sorry, that's right here. But it was not an overwhelming winner either. But there were so many other differences between patients and controls in terms of task effects and the modulation. So we found, again, greater modulation of left MFG on left IFG for PWA versus control. So, emphasizing this point of the importance of left MFG. And we also found higher fMRI task accuracy and semantic performance related to this connection between the left middle temporal gyrus and inferior temporal gyrus, which again is consistent with what we would be expecting to see in this, based on this task. So the bottom line in both these studies is that left MFG is part of a network with left IFG and influences the accurate language processing. And in this case, we're looking at semantic processing and naming. So in the next study, actually, this is Erin Meyer's work. And so in her dissertation, she then said, okay, why are we restricting it to 3 regions? What if we expanded beyond these 3 regions to other parts of the brain? And in this study, we try to develop a more complicated model. Here we're trying to go through stages of the degree of damage. So what you see up here is a normal brain with no damage. It's a control brain. Language performance is normal. You see that there's left MFG in this network. There's IFG, there's MTG, but we're also including angular gyrus. And unlike the previous studies, we have right hemisphere homologs of all of this. And then we develop different model spaces where there's either posterior damage, so the connectivity now shifts to the front, and some to the right hemisphere, or there is anterior damage, so the connectivity shifts more posterior or to the right hemisphere, or pretty much the entire language network's damage except the MFG, and the connectivity shifts completely to the right hemisphere, or you basically have nothing in the left hemisphere and the connectivity is entirely in the right hemisphere, and we expect that these are really the highly impaired patients. So these were 30 patients. They did the same semantic feature verification task that I showed you in the previous slide. They're pressing buttons now about semantic features. And what we found was that while healthy controls demonstrated a preference for left lateralized connected models, which is what we would expect, patients demonstrated a split between the set of models that looked like a control model, and then another set that reflected bilateral posterior weighted connectivity that's subsequent to sort of this anterior damage here. So a lot of, to residual left hemisphere activation, but also some right hemisphere activation. And what we also found, sorry, I'm still on this part here, but we also found that there were several connections between and within left hemisphere and right hemisphere regions that were significantly modulated. So there are more inter-hemispheric connections here. And when we started looking at the degree of damage related to the degree of connectivity, this is a principal component analysis in terms of the connectivity, damage, and performance. What we found was that patients recruited left hemisphere frontal intrahemispheric connections like LIFG to LMFG, inter-hemispheric connections between left and right IFG triangularis, as well as right hemisphere connections. And sorry, I should probably move my right hemisphere connections here. And consistently, what we found here was that greater spared tissue in MF and left MFG and left MTG was associated with stronger intrahemispheric connectivity. So the point here is that, yes, lesion volumes were associated with left reduced frontal connectivity, which is sort of what we were expecting to see. But we also found greater temporal connectivity, which is sort of what this shows, that there will be reorganization of connectivity, and it'll not just all shift to the right hemisphere. There'll be regions in the left hemisphere that can sort of still support that function. But of course, at some point, greater damage to these frontal regions does involve a shift to the right hemisphere, which is what you're seeing there. And this is based on the degree of damage that you see. Okay, so this is, that was looking at fMRI data. We've also tried to look at the patient's brains very differently. Here, we're trying to look at patterns of structural disconnection associated with chronic language impairment. So this data includes 76 participants. They come from different sites that I'll mention. And these guys are getting a language scan, as well as a language assessment, as well as a MRI scan. And what we're trying to do is, sorry, do you have a question?
No, I'm sorry, please go ahead.
So what we're trying to do here is voxel-based disconnectome mapping, and I'll explain that very briefly in the interest of time, but we're trying to look at the correlations between disconnectome maps, which is the probability of disconnection at each white matter voxel, and we correlate that with language scores, and we get a value. And then we also do the traditional voxel-based lesion symptom mapping, where we're just looking at damage to the voxel. So it's not a probability, it's frank binary damage, yes or no. And what we found in these patients when we correlated it with language function was that a common core of white matter perisylvian network was disconnected in 90% of the individuals with aphasia. And surrounding this core is the disconnection in the left temporoparietal network. And this disconnection, or sort of widespread disconnection, is related to syntactic comprehension, is related to syntactic production, as well as to repetition. And what this means is that the disconnection, not just the damage, the disconnection can explain the language impairments, and these extend beyond the white matter lesions. And it's regions that are not even damaged, that are further away from each other, that still can't talk to each other. We didn't find relationships between aphasia severity and auditory comprehension, but I think that might have been a power issue. And so this actually shows the comparison between the VLSM and the VDSM analysis, and I'll probably just skip that for now. So that's all just traditional MRI, fMRI data that we've been analyzing, but we've also taken a different approach towards looking at brain scans when people are doing FNIRS. So this is a different approach. We're actually putting FNIRS caps on our patients, and we're trying to still figure out how to manage the lesion. But the big advantage of this is that we can actually have patients do these tasks without having to go into the scanner, which creates a lot of motion issues for us. And we can also have them do a wider range of tasks than just picture naming and semantic feature repetition. So in the next slide, I'm just going to tell you a very, very early study that we just finished. And here we're looking at patients with aphasia. There are only 6 patients because we're just trying to test the feasibility of this approach, but we've run a bunch of healthy controls. And here, we have the same brain regions we're trying to look at from our fMRI data, but we're doing this in the context of FNIRS. And so we create these probes of regions that we want to look at. We have them do the exact same tasks I've been telling you about: semantic feature, picture naming task, as well as an arithmetic task where they have to do math, hard versus easy. And this is from Fedorenko's lab. And what we've done for our patients is we try to exclude channels where the patient has a frank lesion, so we know where the lesion is, and we turn off those channels. And then for the remaining, we calculate oxygenated hemoglobin and deoxygenated hemoglobin changes. What we see so far, at least, is that healthy controls activate a core language region like left IFG, triangularis opercularis, when they're naming or judging semantic features. That's exactly what we've seen in our fMRI studies. And bilateral domain general regions, when they're such as MFG and SMG, when they're solving hard relative to easy addition problems. Again, exactly what we expect. And so at least the tasks are doing what they're supposed to do. We find that healthy controls, older healthy controls also show similar activation, but they're activating more frontal and domain general ROIs in the semantic feature and arithmetic tasks, and more right hemisphere homologs during the picture naming task, which is not surprising to us because our older adults, will show some right hemisphere change and activation. Our stroke patients is very preliminary data. They're showing activation in the kinds of regions we would expect to see. So for a semantic task, we would expect to see some SFG activation or right IFG triangularis. For picture naming, we'd expect to see middle temporal gyrus. But here we go. It's actually much less activation than we would have expected. And they seem to be generally different than healthy controls. So we're still in the process of figuring out if this is really hypoactivation or it's a power issue. But at least at this point, they're able to do the task and they're able to show the kinds of activation. So we'll expect to see more here. So moving on, everything I was telling you about so far is just looking at neural markers of language impairment. We've also done several studies looking at the relationship between how brains change as a function of treatment. So this is a combination of neural marker and treatment. So in the next few slides, I'll be explaining to you what we see in terms of brain activation and after people receive therapy. So in the first study I want to talk about, it was an fMRI task again. We had 26 patients with their age-matched controls. They're getting fMRI scans and they're also getting a naming treatment. The fMRI scan, in the task involves them to name a picture, just like the one I showed before. So they say hat or they say skip. And now I should tell you a little bit about our treatment because these patients come in for 10 weeks and they practice naming, going through a series of steps that I'll explain a bit more in detail later on. But essentially it's called semantic feature naming treatment because they go through the semantic attributes of a hat and they review the semantic features and they get a lot of practice naming it. So what we saw here was, so we had 26 patients who received treatment. Sorry, that was 12 weeks. And then 10 controls who did not receive treatment. And as we expected, naming therapy on this top graph resulted in a significant increase in cortical activation. This was an effect that was largely driven by patients who responded to treatment. And then we also found that patients who did not respond favorably to treatment had little change in activation over time. But what I think is important is in this slide, where relative to healthy controls, patients had higher pre-treatment activation. And it's all these blue bars that you're seeing on this side in the left hemisphere regions that reduced as a function of treatment. But then there were also in the right hemisphere, lower activation in pre-treatment that resulted in increased activation post-treatment. So first up, this is how complicated activation patterns are when you try to look for change as a function of treatment. Some regions decrease their activation, other regions increase their activation, and you sort of see this relative to what healthy controls show the whole time. So it's not a simple left versus right, up versus down regulation switch. And what was even more important was that when we actually tried to look at the degree of change in these regions relative to the degree of damage in these regions, we found another fairly complicated relationship where in some changes, in some left hemisphere regions, post-treatment changes in activation were associated with greater activation when there was more damage. And in some cases, it was associated with more activation when there was less damage. But I think the important point, the important observation here is that spared tissue in otherwise highly damaged regions can be modulated as a function of treatment, as you can see in some of these here. So the bottom line in this study was there's an upregulation of traditional language areas and their right hemisphere homologs, but spared tissue and improvement are sort of in this nuanced relationship where not every region that's partially damaged is kicking in for recovery. Didn't seem to be that way. So then we started looking at, what is it about these people that are showing these very, very different patterns of activation? Some improve, some don't, some change with an increase, and others don't show a down regulation. We tried to see if there was something about how these patients broke out or broke into groups based on whether they were going to respond to treatment or not. And this responder, non-responder is something I'll come back to later on in the talk quite a bit. But it's the same 26 patients, some combination of that from the previous slide. They're all going through these 12 weeks of treatment, but I'm going to show you data from the semantic feature judgment task, which is we keep alternating between these tasks. It's the exact same treatment. And what we did here was based on their response to treatment that I'll explain to you in a later slide. We had participants either be classified into responders or non-responders. And then we looked at global network-wide measures and local regional graph theoretical measures of pre-treatment functional connectivity. So we looked at, so all the data I'm going to show you are patients' connectivity measures before they start treatment. And we're trying to see if there are differences between their brains, depending on whether they're responders and non-responders. And what I'm going to show you in the next graph here is to what extent that those network measures are predicting naming improvements. And what we found here was, so the contrast here is responder versus non-responder. And the values tell you either node strength, global efficiency, or local efficiency. And responders had a higher level of global integration, that's higher network strength and global efficiency than non-responders. And these measures predicted treatment accuracy, the extent to which they were going to improve in treatment, even after we controlled for lesion volume and age. So this is not about just more severe patients or just worse off because they're not going to improve because they have large lesions. It's really more nuanced than that. And I'm going to come back to this point again and again because this paper was just telling us, hey, there's something different about these patients' brain before they even start treatment. So the extent to which they can improve in treatment is limited. And so this was the first time we sort of made the point that the relative preservation of global and to a lesser extent local efficiency might actually be a biomarker for treatment-related recovery. So we're still going at this work because there's something here to keep looking at. This is another paper. In this paper, it's again looking at these a subset of these patients. In this subset, there's actually 20 patients who receive treatment, and then there's 10 who don't get treatment, and then there's a bunch of healthy controls. And it's the same task, so I don't need to explain this again, and it's the same treatment. But what we did in this study was all those 30 patients, the 20 who received therapy and the 10 who didn't, were compared to 17 healthy controls to identify the presence and the extent of abnormal connectivity among patients, even before any treatment was administered. So this is pre-treatment performance, and we're comparing all the patients to the healthy controls. And this analysis shows that patients who had abnormally low functional connectivity in a subset of 31 connections. So we looked at all the regions of interest, and this is where these patients are just worse than controls. They're never actually better than controls, so there's never a positive effect. These are all they're worse than the control patients. So we call this the abnormal connectome. Then we looked at these regions and we divided the patients, as I said before, to responders and non-responders. And so we had three groups to compare it with. We had the responder group, the patients who ended up improving in treatment. We had the non-responder group who we know did not improve in treatment. And then we had the untreated group, which is the line in red, who didn't receive any therapy. So they should be the ones who remain the same because they didn't receive any therapy. The responders are the ones who actually change in their connectivity in these regions. They actually improve in their connectivity in these so-called abnormal networks. And more importantly, the non-responders actually get worse. They don't show a strengthening of connectivity in this abnormal network. So these results suggest that successful naming therapy is associated with increased connectivity in brain regions that are functionally abnormal before even the therapy started, and the capacity to enhance the strength of such connections may be advantageous. So successful connectivity is associated with increased connectivity between connections that were abnormal, but they're able to change, and there's something about this ability to change that makes these people responders in treatment. So all this is sort of getting us to a theory of what we think is happening in aphasia. And so I'll just tell you where we are right now. This is by no means the end of the story. This is sort of an interim summary of where we are. And we've based our theory of recovery from an earlier paper by Heiss and Thiel back in 2006, where we think there's a hierarchy of recovery with increased damage to the left hemisphere. So there's obviously, I don't think I need to convince you right now that there's a network of regions that are activated in the service of language recovery. I think everything that I showed today was a network of a bunch of regions. But to the extent to which left hemisphere regions are spared, they're important and they're engaged in language recovery. That's what I've been showing you all along, is there are consistently left hemisphere regions that keep showing up in these fMRI tasks. But as damage increases in the left hemisphere, there's this shift towards the right hemisphere, and more importantly, to multiple demand domain general regions, such as the MFG. Was not actually a region we were thinking too much about, but it turns out to be an important region for recovery. And then finally, I think where we are right now is the degree to which this network typology instantiates really determines the extent of this recovery. So it's again not as simple as I'm showing you in this slide here, in the schema here. It sounds like it's all or none here, but it's actually much more nuanced. So I want to come back to this proposed model that I showed you up front, and you were sort of wondering what this was about. But here, what we're really thinking is that there's multiple things going on in terms of recovery. So back to this, this picture here, where this is sort of the time on an axis, days, weeks, months, and years after a stroke. Somebody has a stroke. Of course, there's a decrease in function. I showed you that earlier, but there's recovery that's happening slowly for these individuals. And the line trajectories here really reflect the connectivity or the network properties. So here, it's not activation, it's actually connectivity of the network. And healthy people have this perfectly connected network where everything is functioning efficiently. And stroke patients have essentially a completely disrupted network, which we know about. Now, in the acute stage post-stroke, behavioral deficits coincide with the abnormal functional connectivity and network topology. They're very different from healthy controls, but in the following months, gradual restoration of connectivity and topology coincides with this improvement as indicated by these lines. But when patients reach the chronic stage, they really need treatment. And at this point, in many of them, this abnormal connectivity and network topology persists. So when we give them therapy, patients whose network properties are already higher, already well connected, or at least look more like healthy controls, have a favorable behavioral response to treatment and further normalization. So they get closer and closer to what healthy controls would look like. But those who have these abnormal networks at the start of treatment, even before we give them the treatment, just end up having poorer behavioral outcome and less substantial changes. So like these responders can be distinguished from the non-responders based on their differences in properties. And so we think these are meaningful predictors of recovery, but we're still really trying to get at this question, and you'll hear a lot more about this work in the next few years. So on to the other aspects of what we're putting into our predictive modeling. I'll spend the remaining 10 minutes of my talk going through that. I'm going to skip this slide about language and cognitive markers because we've actually done a lot of work in this space, and I don't have time to go through it. Likewise, I'm going to skip through a lot of the effective treatments we've been developing. Sort of long story short, we've essentially developed treatments for patients who have trouble reading, producing nouns, producing verbs, functional communication, sentence processing. And more recently, we've been trying to do all this in the context of bilingual patients. They're either Spanish English speakers or Mandarin English speakers and so forth. So we know exactly what treatments work. We just don't know yet who they work for. And also, I think I'll skip through this. Actually, I'll tell you a little bit about this study because I talked so much about responders and non-responders. And so here we're training patients to name either typical examples like hat or shirt, or atypical examples like suspenders and apron. That's what the treatment they're getting. And they, as I said before, they go through the semantic feature training by thinking about the features of a hat versus a shirt versus a suspender. And our theory is that when you train the atypical examples, you get more generalization than when you train the typical examples. But what I think is worth noting about here in the context of treatment is when we looked at the whole sample, yes, it looks like we get the training effect and the generalization effect, but really, the results are really driven by these people who are called responders, and the non-responders really just don't respond to treatment, even though they get 12 weeks of treatment. So that's why we're calling these people non-responders, because they're not getting any benefit. And when we look at what examples they're being trained on, again, the people who respond to treatment show the effects, and the non-responders essentially don't show any benefit from treatment. So it's not just the practice that can make a difference here. So anyway, this slide puts in context a lot of what I was telling you earlier about responders versus non-responders. I'm going to try to not talk too much about the next treatment, other than it's a bilingual treatment. We're looking for generalization from single words and when they're trained in English or Spanish. I'll come back to this because I'm going to explain this in much more detail in our predictive modeling slide, but I just want you to know that the treatment that we provide these sort of patients, even though they're bilingual, is very similar. We still have them name pictures and go through the semantic attributes and try to produce them in sentences and so forth. So I'll skip the treatment data because it's not that interesting. What I want to say is that we have spent a lot of time trying to figure out which treatment works for which patient, a reading treatment versus a naming treatment. We've also tried to standardize the intervention for different patients. And the way we've done that is by delivering therapy through computers, because as you know, in COVID, we're able to actually reach out to many more patients as a function of the technology. So this is where I sort of want to land with the last few slides. All of this has really given us a wealth of information to build predictive modeling data sets, and that really is getting us closer to better treatments and prognosis for our patients. We have not been able to do that with a single small studies, but we can do it now with these large data sets. I'll give you 3 examples of how we're doing this. In the first case, we're actually looking at really large sets of data, and then progressively looking at smaller cohorts down to the individual patient. This is the first example I'll talk about. We're doing this in the context of data we've been able to get from Constant Therapy Health, which is a software program that I helped develop. It's a commercial software program. Patients can download this app and practice therapy at home, but that's given us really, really rich data for patients practicing any kind of therapy on their tablet, and we calculate accuracy and reaction times. So these are just examples of the therapy tasks, and we've published a lot of papers on this already. The reason we're able to do this is because pretty much everybody who has had a stroke and has been to a hospital is practicing these therapies from their, as they're being prescribed to do it in the hospital and across the world. And that's given us the ability to really tease out factors that we need large data sets to be able to do. So the factors such as, how much therapy, how much dosage, what type of therapy are questions we could not answer before, but we're able to answer those sort of questions now. So in this first study, in this first study, I want to talk about, I will talk about optimal dosage. So how much therapy does somebody need to improve? And the question we're trying to ask here is how much therapy dosage does a patient need to change their performance? And we can do this because patients are doing all this therapy at home. So we take data from about thousands of patients and we narrow it down to these 2,200 patients who've all had post-stroke speech and language impairments. They've used the app for about 10 weeks. And we can then aggregate the data based on specific skill domains. So it could be auditory comprehension or phonological processing or reading, because we have that much data. And then we try to analyze the degree of improvement based on chronicity by accounting for age, chronicity, baseline severity, and dosage group. And what's important here on this graph is the amount of dosage. So everybody's receiving 10 weeks of therapy, but there are patients who are only getting therapy once a week. Others are getting it twice a week, thrice a week, and there are other patients who are practicing 4 or 5 times a week. And I should say that these are all patients practicing at home on their own on the app. And as you would, not surprisingly, see that more practice is better. So the 4 or 5 times a week practice is way better than practicing once or twice a week in this data set. But what's even more important is that that the degree of improvement that someone can make is based on how much practice they do. So if people only practice once a week, the rate of change is much lower than patients who practice multiple times a week. So they actually can get better more if they practice more therapy, which is something that clinicians sort of intuitively knew all along, but they've never known, this data actually proves that you can actually do this. Okay, so let me move on to the other two examples of what we're trying to do with predictive modeling. These are very, very different types of studies. In the first study, we're trying to look at neural imaging markers and behavioral markers that predict treatment outcomes. So they're both predictive modeling, but they're very different. In this data set, we have 55 patients with aphasia. We are looking at their neural markers, all their imaging markers, but also we're looking at behavioral, how old they are or how severe their language is. And here we have factors such as age, months post-stroke, their aphasia quotient, how severe they are, their cognitive scores, and then a host of neuroimaging measures such as how large their lesion is, how much spare tissue they have, how much damage there is in the white matter regions, and then even connectivity between regions using the resting state fMRI data. All of these go as input variables, and what we're really trying to be able to predict or classify is whether somebody is going to be a responder or non-responder. And in this data, this is 55 patients. It's a much larger data set that I've been showing you before. And the criteria we've set here is just 25% or more improvement. We're using two sort of models here, SVM and support vector machine and random forest models. And we rank order how good or how well these features can predict treatment outcomes. And this is all work that's going on right now, but right away, what we find is that both models can fairly accurately predict who's going to be a responder or non-responder. But the way they do it is by taking multiple sets of these features. No one set of features is really a unique winner, although resting state connectivity is actually a very consistent measure of recovery prediction, as is aphasia severity. So that's data from the support vector machine model. It's something that's, the results look somewhat similar for the random forest model. It's still resting state and aphasia quotient, but the model results aren't as great. Again, this is ongoing work, but at least it tells us that multimodal information, especially something like resting state connectivity, can get us far. And then the last predictive model I want to talk about is a computational model, which is very different. This is a neural network simulation of a bilingual language system where we create a semantic map where it can process semantic features of dog, but then it either is able to produce the English word dog or it produces the Spanish word perro. And what we've done in the past is we've simulated bilingual aphasia in this neural network model. We've simulated language recovery in this bilingual model. So we actually treat it like it was a patient and have it recover language function. So we've already done all this work and shown that we can, this computational model does a pretty good job simulating bilingual aphasia. What's really important and interesting right now is we have now taken that, a couple of steps ahead in terms of a randomized double-blind clinical trial where we're assigning patients to a treated language or the untreated language based entirely on the model's predictions. So we give the model the data of the patient beforehand. It simulates these outcomes that I'm showing you here. And based on those outcomes, it tells us you need to train the patient in English or in Spanish. And then that's the prescription recommendation for the patient. And we actually train the patient either in the model prescribed language or the model opposite language. So there's a whole clinical trial that we're pretty much wrapping up at this point, where the patient either receives therapy based on the model's prediction or opposite to the model's prediction. And I'm really excited about what we'll end up finding here because this is really a game changer for aphasia rehabilitation. So stay tuned for those results. And I think whoops, I think
You're fine. You can continue if you want.
yeah, actually, I'm in my last slide. Let's hope it doesn't crash. Uh, Oopsie. Let me stop share for a second and restart. I think it's sort of, but I'm happy to take questions while I wrap up my slide as well.
Sure. Okay, so we have our first question from Yifan. Yifan, can you please unmute?
Okay, yeah, can you hear me okay?
Yeah.
Yes.
Okay, yeah, thank you so much. It's excellent work. Really amazing result, very interesting to see. I have a very quick question. It looks like on your one of the figure indicating that a few months after a stroke, um, uh, you started the treatment and the people with a strong, neural connection to other regions of the brain seem to help them to respond to the treatment. I'm wondering if you or others have looked into how early the treatment can be administered and that would be more effective. Is there any sort of study to show, is earlier is better, or is any particular timeline that will be more effective? Thank you.
Yeah, that's a great question. Actually, there's a lot of data that shows that early treatment is better, but very, very early treatment is not very good. So if you give patients therapy in the first 1 week and it's very intensive, you can actually do more harm. And that's actually what motor recovery studies have also shown, is that you're not necessarily helping them. But in that subacute phase of 2 weeks, 3 weeks, 4 weeks, patients show a tremendous amount of spontaneous recovery. And most studies that have tried to compare intensive therapy during that time have found, relative to usual care, have found it to be beneficial. And the gains are really significant. So there is a clear advantage in doing early therapy. But I think the message that we are trying to dispel, because this is a huge missed message, is that only if you do therapy early does the patient recover. And once you miss a certain point, there's no more recovery happening. And that's why we focus on the chronic stage, and all this data is showing that even in the chronic stage, patients will recover function. And that's very important to show as well.
Thank you.
Great. We have another question from Tamar. Tamar, can you please unmute?
Yeah, hi, Swathi.
Hi there.
So actually I asked, where do you see the field in a few years? Like, what treatment you think will be individualized, and also what treatment do you predict substantial changes in the methods of treatment, or will it remain like traditional treatment of naming, etc.?
Yeah, no, that's a really good question and a really tough question, actually. I do think that treatments will get more individualized. I think people will say, I need to, actually, that's what the slide is about, is that I think for some patients will say, you need a boot camp. You need to go to a facial therapy for 6 weeks, every day, day in and day out. And whereas other people probably need social communication treatment because that's what they want to do. But right now, it's all trial and error. I think we'll have enough data in the future to be able to tell which person what to do. And in that case, I actually feel like it's a little sad, but I don't think the kind of treatment we do, whether it's VNest or naming or sentence production, will matter as much. It just won't matter because you're training the brain, giving some intervention. So I think that's good news, bad news, because what specific intervention you give may not matter. The fact that you're giving that intervention over long periods of time or in a functional context will be really important. But I think that's good news for the patient, because that means that they will actually get what they need in the long run to communicate.
Thanks.
Great. And there's another question from an anonymous attendee. How do we understand the role of thalamocortical connections when it comes to recovery outcomes? Do you suspect these connections may play a role for responders versus non-responders?
Yeah, no, that's a really excellent question. So of course, everything that I showed you so far is about the the cortical connections, but I'll just go back, if I can go back to this. The only place where we can actually test this is in this model that I just showed you. Oh, oops, no, too far ahead. So in this computational model, we actually think we can build, in this model, we think we can build a basal ganglia corticothalamic circuit into this, because we can try to do it here. Everywhere else, we only have what the patients give us by way of lesion. But in this place, we can actually try to build that in and see how it interfaces with the cortex, especially in this case, maybe the temporal lobe, and how that relationship works. And we think that there's a huge gating mechanism that's at play that we're not even capturing right now in terms of recovery and attention and so forth, that we'll probably be able to study more carefully.
Thank you. Bruce Jenkins, can you please unmute?
Hi, yeah, that was a fabulous talk. A little hard to follow it all, so it's very dense. But what I was curious about is, most of these subjects that you study have, damage over a pretty widespread network, throughout the left hemisphere. And so what I was wondering is, have you looked beyond, like, instead of just looking at language-related areas? I know you're in aphasia, so of course that's where you're focused. But are there relationships between networks for other systems, specifically, like, say, for motor systems and for other cognitive domains that might have been affected by these? And can you actually start to develop inferences about changes in motor systems, for instance, helping or hurting? Because when you're trying to do a task in the magnet, it's some of these tasks rely upon motor skills. So there's other pretty basic other networks that might be related with what goes on in the aphasia networks, especially if you're talking about global connectivities. One would think that you could actually start to build kind of a bigger picture of how these are interacting with the ability to recover.
Yeah, no, that's a great question. That's exactly what we're starting to do now. I mean, I think it makes a lot of sense for somebody outside the field to say, well, stop looking only at language network, look beyond that. But it's taken our field a while to say this is more than just what's happening in the language network. We need to look beyond. So I think we're all finally there now, and I think we're able to look beyond. And so our work is really trying to look at the default mode network and the salience network, and to a certain extent, we're not really looking at the motor network, but we could. And we're trying to see how those networks fit with the language network. And Brenda Rapp, who's part of our research group, has a paper that came out that also started looking at how these other networks connect with the language network. But now is the time to start doing that, because I think we're all comfortable that there's other things going on in non-language regions that are contributing to language recovery. And so we're starting to look at that now. That's a great, hopefully in a year or two, we'll have more data on that.
Great, thank you very much. So I think that concludes our Q&A phase. So I'd like to thank you very much, Professor Kiran, for coming here today. And we hope to see you again in your third BrainMap talk soon.
Yeah, thank you very much. Thanks, everybody.
Bye, everyone.